Hi everyone, my name is Jess. I'm super excited to be presenting for the ASFSN this afternoon to close out the data blitzes. So I'm going to be talking about one of my PhD studies. So we all know that social cognition is really important in our everyday lives. So being able to recognize someone's emotion, understand how they're feeling, and act appropriately in social settings is really key to all of our interactions. So these abilities, as we've just heard, can be affected in neurodegenerative syndromes. So in Alzheimer's disease, we see subtle changes, particularly in these more complex social situations, and these usually occur later in the disease. Behavioural-variant frontotemporal dementia, on the other hand, present with profound deficits in social cognition, occurring quite early. Parkinson's disease is another neurodegenerative syndrome. We usually think of this as a movement disorder. However, impaired emotion, recognition and empathy has also been observed in these syndromes. So a large amount of the research to date has focused on how these patients process external cues and care-rated questionnaires. So less is known about how these patients process this internal information from their body. So interoception is the process by which we receive and interpret physiological cues from the body, and this is tied to emotion. So you might notice these cues, for example, when you're excited or afraid or scared or public speaking, you get an influx of this physiological response. So a difficulty in interpreting these bodily cues might contribute to the impaired social cognition that we see in dementia. So on this figure here are some of the brain regions that are implicated in interoceptive processing. So we can see that there's some lower level brainstem, subcortical and frontal regions. And the reason we're interested in looking at how interoception might be affected in dementia is that we see an overlapping pattern of atrophy within the different disorders. So typically research to date has focused on cardiac interoceptive accuracy, which is the ability to accurately detect your own heartbeat. And this involves the insula as well. So only a handful of studies to date have investigated cardiac interoceptive accuracy across these syndromes. And there's been some conflicting evidence. So some have found impaired cardiac interoception, while others have reported intact. And also a lot of the studies to date have used regions of interest and not whole brain atrophy. So we aim to investigate cardiac interoception in a large neurodegenerative sample and the associated neural correlates, hypothesizing that all patients would perform significantly worse than controls and that lower performance would be associated with these brain regions on the screen. So to do this, we recruited participants from Argentina, Australia and Chile, and all participants completed two 2-minute tasks. So simultaneous ECG was recorded, and during the extraception task, they pressed a button each time they heard a recorded heartbeat. Whereas in the interoception task, they pressed a button each time they felt their own heartbeat. We then calculated a measure of accuracy. So this compared the frequency of the event in each task with the frequency of the participant's response, with a lower index meaning better performance. We also looked at neuroimaging, so this was structural MRI to conduct VBM analyses. So what I found was that the Parkinson's disease patients were worse than healthy controls on extraception, whereas on interoception, everyone found the interoception task harder. However, BVFTD were worse than controls. And then when we looked at the neuroimaging correlates, we found that a number of significant clusters in these subcortical as well as cortical regions, and these regions have been known to be affected involved in interoception and arousal. However, they're also crucial for emotion, reward processing, learning and memory. So given this overlap between functions, my next step will be to investigate the associations between interoception, emotion, and memory. Big thanks to my supervisors, my collaborators, the Frontier team, the patients and their families, without whom this research would not be possible.